It's okay. Welcome, everyone. Thank you for being with us today. We're broadcasting live from Park City, Utah and San Diego, California, right, Farhad?
Actually, Santa Monica, California.
Santa Monica. That's even better.
It is.
Yeah. So we're grateful that you're with us today. Big hug and thank you to Jason Rifkin, who I just noticed came on, for introducing us to Farhad and Fahad. Thank you, Jason. Super interesting subject matter here, especially if you're focused in this sort of thing. As you're going to discover, Farhad is not just kind of a rock star, but a rock star in this silo of advanced therapeutics. Already way over my skis, but I've been impressed not only with the discussions we've had, but you're going to soon find out that the people who are in the know have been with their checkbook in terms of not just capital, but non-dilutive capital. So I know you have a couple of announcements to make too. So we'll look forward to that. Again, thank you for supporting Family Office Insights and regularly participating and engaging as you have. A couple of housekeeping items here with Zoom. As you know, we can't hear or see you, but we welcome and encouraging questions in the chat box or the Q&A, and we'll be monitoring them. Please put them in as they come to mind so we can get to them in order and as many as we can. I'm also going to be recording this, and with Farhad's permission, put it on the YouTube channel, our Family Office Insights, and so you can review it there. And also feel free to, if there's others that you think would benefit from knowing what you learned today, you can send them over there. And of course, we'll also put you in direct touch with Farhad. So with that, again, big thank you, Jason, for the introduction. Not only is Farhad clever, I've come to like him, and I don't like everybody. So, and I'm not too hard to get along with. So, so with that, again, thank you all for being here today. Take it away, Farhad.
All right. Well, first of all, I have to give my gratitudes to Arthur and his excellent team at Family Office Insights for the work that they did to provide this opportunity for me to present MAX BioPharma today. And it's really exciting when I get to share what we do at MAX BioPharma with an audience, because my hope is to really introduce groundbreaking science and discoveries that have happened at MAX BioPharma and show you how far we have come in really a very brief period of time that we've been in existence, and how we have and will continue to impact the way that therapies are discovered for a number of human diseases. So thank you, Arthur, and thanks to your team. And of course, thanks to my friend Jason for introducing us and for being here today. Just before I get into the meat of the discussion, let me give you just a little bit of background on myself so you know who I am and where I come from. So I was trained as a molecular cellular pathologist at UCLA with a Ph.D. degree. Prior to that, I had a Bachelor of Science degree from UCLA and eventually was placed in the M.B.A. program at UCLA again. So true Bruin here. After having spent about 22 years as faculty at UCLA in the Department of Medicine, I decided that in order to really bring our discoveries that were made during that 2 decades into fruition and into making a difference in how patients are treated, I had to form a company. So the company, actually MAX BioPharma, was founded in October of 2010, and we remained virtual, really working out of my lab at UCLA for about 3 years, after which we got an independent space at the Magnifying Incubator at UCLA, which was really great for us. It facilitated our ability to start discovering even newer things that than what we had discovered already at UCLA. And then after 3 years, starting in 2017, we obtained our independent lab space here in Santa Monica. And we have been here and we've been quite productive despite the 2 years that we had of COVID that really disabled a little bit of our activities, but we are back and we are doing very well. So on that note, if it is okay with Arthur, I will start the slide deck. Just another piece of information that might be interesting for some of you is I often get question about why MAX BioPharma, and let me just give a little background on that. So my late father was Benjamin Parhami, and he was a pharmacist in Iran and started one of the first pharmaceutical companies in Iran in 1963 by the name of MAX Laboratories. And unfortunately, after the revolution, he had to leave everything behind and come out. And when I started MAX BioPharma, I thought that this would be a good name for a company that now I have founded here in the United States as a legacy to him. So that's why MAX BioPharma, just in case. But of course, MAX also abbreviates molecules for advanced therapeutics. So just a little there.
Double entendre sort of thing. Yeah, nice. No, yeah, please carry on.
Okay, great. So as I mentioned to you, MAX BioPharma really originated at UCLA, and this was based on at least 2 decades of work that my team and I had done in order to study a class of compounds called lipids. Lipids are basically fats, and they come in all sorts of varieties. And what we discovered was that, which was pretty groundbreaking at the time, was that there are very specific lipids in the body that can regulate certain biological activities that previously were not known to be activated or modulated by these compounds. So these compounds collectively are called oxysterols, and I'll get into this more later, but based on one compound, Oxy 133, that is now a lead compound for us in and one of our most advanced therapeutic development programs, became the meat, if you will, for MAX BioPharma to begin its work. And since then, and since we became independent of UCLA, we have also created new and independent IP, which has really brought us to where we are now. We continuously go after our initiatives, which have been and continue to be performing preclinical studies in order to identify new novel candidates for therapeutic development based on our Oxysterol Therapeutics platform, which I will get into later. And to eventually file with the FDA and start clinical development of our drug candidates and continuously expand our intellectual property, which obviously creates a lot of value for the company. Our funding has come almost entirely from non-dilutive government grants of now more than $8 million. Plus a small seed funding, and of course, family and friends who helped us in the beginning. And we continuously, again, rely on government grants, and we have been rather successful in bringing in a number of SBIR grants and more to support our activities. So this is really the first time that MAX BioPharma is raising institutional money in order to support the further advancement of our work. We, again, are always looking for strategic partners who can bring in scientific as well as technical and financial support to our activities, and those are either in the form of joint development projects, or we're also looking to out-license some of our intellectual property. So this really has and will continue to maximize the value that Max BioPharma brings to present and future investors. So our strategy has really focused on exploring a previously underexplored chemical space of naturally occurring and semi-synthetic proprietary sterols and oxysterols. Now, oxysterols were always known to be part of biology, but their candidacy as being a potential therapeutic candidates only came into fruition when we stepped into the field. So we've been focusing in this area, and it has been a very good starting point for us with a reasonable rate of success that you will see. And we have been able to identify high quality drug development candidates for a number of diseases and indications. So this entire work that we have done has created the opportunity to build what we call an Oxysterol Therapeutics platform technology. And that's shown here in the center of this diagram. And the Oxysterol Therapeutics is basically now a library of small molecule oxysterols that are more than 300 and ever expanding. And we have been able to screen and study these oxysterols and come up with a number of therapeutic candidates for several diseases and indications. You can see here we have successfully gotten into the area of bone formation or stimulation of bone formation. There are many diseases that require an increase in the amount of bone formation by the body, and that's where we are focusing. And with our lowest hanging fruit is in this area of spine fusion, which is, I will get into more later, but it's basically stimulating localized formation of bone in order to help heal the body. We have also been able to identify novel compounds in the areas of fibrosis, fatty liver, just to name one, which is our focus at this time, and also for cancer, including lung cancer and pancreatic cancer, which, as you know, is a very deadly disease still, despite all of the advances that has been made in treating pancreatic cancer. We've also dove into viral infections, namely COVID-19 and hepatitis B virus, in collaboration with colleagues at the National Institute of Infectious Diseases in Japan, in Tokyo, and more recently with the NIH, where we have identified compounds that are inhibitors of viral infection in COVID-19 and in hepatitis B virus. And there are multiple other indications, and also I can't leave off chronic inflammation, which is a new area that we have really gotten into recently because of new discoveries that we have made at Max BioPharma. So this is our platform technology. It's really very robust, and it will eventually lead into many different avenues that will help solve a lot of the unsolved medical problems that are currently existing.
So, can I interrupt 1 second?
Yes.
Can you jump back to the one before that?
This one?
The one before this one? The one even before that, sorry.
Okay.
I just wanted, because I think others might be thinking the same thing, is this seems like there's a lot of vote of confidence from non-dilutive capital and grants. Is this an unusually high amount of grants compared to your overall funding for a company like yours?
I believe so, especially for a startup company. For those that are familiar with NIH funding, it is never a guarantee. It is a very challenging process, because when you put in an application, it has to be scientifically foolproof, and it also has to convince your peer reviewers that the science is correct, and because this is an SBIR, that it actually fits with your business strategy, and it will actually be a fruitful contribution to the success of a company, for example, Max BioPharma. So yes, so having raised as much as $8 million in non-dilutive funding is pretty significant, and we're very happy about it.
And how long does each one of those take about? You don't need to be precise, even though I know you're a scientist. Is it 10 months to get one, or is it 18?
Well, right now, the trend is that you put in an application, it gets reviewed, and it almost always comes back with suggestions and comments that you have to incorporate. And that is if you receive a score. Sometimes your application is unfortunately triaged, and we haven't had that many of those, but it takes at least a year to 18 months to get one of these grants. But it is non-dilutive funding, so it has been our bread and butter and our support so far.
Yeah, super interesting. Thanks for
of course
clearing that up.
Thanks. So so the next slide, which is a Gantt chart, shows you our different programs, and this is really divided into our primary focus as well as other opportunities. And really, as a company, as a small company, we had to make a lot of hard decisions on what to focus on and how to spend our very valuable and hard to get financial support that, as you heard, comes entirely from NIH grants so far. And based on the data that we had, and based on the trajectory of how difficult or simple is going to be to get through the FDA clinical trials, and how big the unmet medical need is in the spaces that we're involved in, we decided to focus on two areas. One is using our lead compound, Oxy-133. Again, it's an oxysterol compound that is a very potent stimulator of bone formation. It targets stem cells in the body to make them become osteoblast bone forming cells. And what we do is we take this and we apply it locally in a spine fusion indication. And I'll show you some slides of spine fusion for more granularity later. And this compound was discovered at UCLA, and we obtained global exclusive rights from UCLA in order to develop Oxy-133. So spine fusion is our most advanced program so far. Currently, we are supported by a $1.7 million grant from the government that is allowing us to perform IND-enabling studies in order to get to the FDA filing stage, hopefully in the next 12 to 18 months. And the second area that we're focusing on is a disease that is involved with liver, and that is NASH, non-alcoholic steatohepatitis. This is when the liver starts to accumulate fat and becomes inflammatory, and unfortunately, it is a disease that progresses, and there is absolutely no drugs for it currently. And we have selected a lead compound from our Oxysterol platform technology called Oxy-210 that blocks multiple pathways that are involved in the development of NASH. And we have been supported by a phase 1 SBIR SBIR grant that has allowed us to come so far in the development process. And actually tomorrow, we have a very substantial phase 2 SBIR grant that's going to be reviewed in order to allow for the start of IND-enabling studies for Oxy-210 to target NASH. So this is independent IP that we developed in Max BioPharma. And there are multiple other opportunities that we have actually been involved with in the areas of other fibrotic diseases, such as pulmonary fibrosis and kidney fibrosis. In the area of cancer, oncology, we have had publications on pancreatic cancer and non-small cell lung cancer in collaboration with our colleagues at the National Cancer Institute. As I mentioned briefly, we are also have explored the area of oxysterols for viral diseases, HPV and COVID-19. For osteoporosis, we are hoping to take our Oxy-133 and make it bone targeted as an anabolic agent for the treatment of osteoporosis, whereas probably many of you know, requires stimulation of bone formation that the body has lost during aging or after menopause in women. And also in the area of inflammation, where we have now found very potent anti-inflammatory oxysterols that we hope to develop for diseases that involve chronic inflammation. And all of this, again, is based on IP that we have developed independently at MAX BioPharma. So just to give you a little bit of granularity at the problems that we're trying to solve, this is a slide that shows spine fusion. So some of you may have seen slides like this, but a lot of times there is a need for correcting spinal instability. And that's when orthopedic surgeons or neurosurgeons go in and they fuse multiple vertebrae together using these rods and screws, but also what is used is a biologic called Infuse or recombinant human BMP2, which is a product by Medtronic. And what it does is that it stimulates the growth of bone, formation of bone that bridges the multiple vertebrae that require to be affixed together in order to give stability. Unfortunately, Infuse in the past decade has run into a lot of problems in terms of its safety and a lot of controversy about how it affects patients and patients' health. And as a result, its sales have really plummeted from more than 800 million, or actually $1 billion, to less than 500 million now. So there is a huge need for having a biologic that is going to increase the chances of fusion to occur completely and giving the necessary stability to the spine of a patient. This is, again, a rather big market. There are about 1.6 million annual instrumental spine procedures that are done in the United States. And this includes 352 interbody fusions where multiple vertebrae are, in fact, fused together. So this is spine fusion. This is the problem. And the way that we would like to solve this, and we propose to solve this, is using our Oxy-133 lead compound in order to stimulate robust bone formation. And we always compare our compound to Infuse, which is, again, the gold standard. It has been the gold standard because of how potent it is in creating bone where bone is needed. And what we have shown is that Oxy-133 is, in fact, as efficacious, as potent as recombinant human BMP2, while being safer and much less expensive to manufacture. And I will show you a few data slides just to tell you what it is that we're comparing ourselves to. But the way that it works is that Oxy-133 targets very specialized stem cells in the body called mesenchymal stem cells. And these cells are basically progenitors of bone-forming cells called osteoblasts. And we make them realize that, okay, now you have to become an osteoblast and make bone. And these have basically been shown in vitro and in vivo in cell culture and also in animal models to be very potent stimulators of bone formation. And these osteoblasts mature and they make a lot of bone. So we actually have published a number of peer-reviewed papers on Oxy-133 together with collaborators in multiple institutes, including at UCLA and USC and Johns Hopkins. And this is only 5 of those peer-reviewed papers that appear here on this slide, just so you see that this has been validated many, many times. Yes, go ahead.
If it's okay with you,
Yes.
we have a good friend, Stellar Human, by the way, who's on the call that has to leave before you're going to get to some stuff with a series of questions. Can I pose them to you now?
Absolutely.
Okay. So our good friend Pete, thank you, Pete, for being here. There's questions around the IP. Do you own it or license it from UCLA? I guess that's the platform question, right? And if it's licensed, does UCLA have an equity interest?
So the only IP that we have that's licensed exclusively from UCLA is Oxy-133, which is being used for spine fusion. Everything else is fully owned by MAX BioPharma. And as part of the licensing agreement with UCLA, there's no equity involved.
Great. And then finally, is there active strategy that you can talk about or suggest that you might with regard to working with strategics such as J&J, and I guess EG is another one?
Yes, so we have actually had multiple discussions with multiple of the big players, including J&J and Stryker, who are waiting for us to develop the program to a point that they feel is de-risked enough that they would want to jump on and acquire it. But just to give a very small amount of background on where Oxy-133 has been, Oxy-133 was actually sub-licensed at one point by MAX BioPharma to Medtronic. And this was in their hands for about 2 years, but unfortunately that collaboration ended when the old guard that had signed a licensing deal with us left, and the development of the compound started to not go as we had planned. And that's why the agreement was terminated. So just the fact that Medtronic did their due diligence and picked up Oxy-133 to develop alongside their rather successful product, even despite the fact that it had all the safety issues infused, is testament to how important Oxy-133 is. And if you talk to any orthopedic surgeon or neurosurgeon who performs spine fusion, you will see the significance of having a product like Oxy-133 in the spine market, but also for other localized bone formation indications.
Great, super helpful. Thank you for doing that.
Great, thank you. And thanks, Pete, for the questions. So this is just one example of many that we have published. And this is a rat spine fusion model. And as you can see on this side that I'm pointing to, this is a rat, a control rat that has its vertebrae separated, as you can see by the empty spaces of the arrowheads. And when you implant, infuse, or recombinant human BMP2, you can fuse multiple vertebrae, as you can see here, by this robust amount of bridge of bone that is formed. So that is, this is successful spine fusion, where you actually have a complete fusion of multiple vertebrae, and you lose basically the ability to move these vertebrae individually. But that's the reason why spine fusion is done, is to give stability where stability is needed in the spine. So this is with recombinant human BMP2. And as you can see, when we apply Oxy-133, our lead compound, at 2 different doses, you get as much of a bone formation and bridging as you see with recombinant human BMP2, while the quality of bone being superior to that of BMP2. And this is in our publications. But in this rat study, we showed that even at a low dose of 2 milligrams, you can see robust fusion, and at 20 milligrams of this compound, you get 100% fusion as seen by X-ray and by micro-CT. So this is work that was performed by Dr. Jeffrey Wong at then UCLA, now at USC. And it basically gives you proof of concept that the Oxy-133 is not only in vitro a potent osteogenic factor, but it also is a very potent bone-forming factor, osteogenic factor, when implanted in vivo in a preclinical model of spine fusion. We also performed this in a larger model. This is a rabbit model. And again, you can see that in this rabbit model, in the control vertebrae, where the arrowheads are, there's a gap. So this is where the fusion needs to be. And when you apply recombinant human BMP2, you get a very robust amount of bone formation that fuses the vertebrae. And with Oxy-133 at 2 different doses, you can see the amount, the volume of bone and the connectivity of bone that is causing a bridge between the vertebrae that has caused 100% fusion. So we have repeated this study in multiple different settings, but also our collaborators at UCLA have shown that not only spine fusion, but also other indications such as cleft palate can be reversed by Oxy-133, where bone is formed. Also in maxillofacial and cranial defect models of rabbits, scientists at UCLA have shown that Oxy-133 is a very robust bone-forming agent that can help regenerate bone where bone is needed in multiple areas. So this is our spine fusion model and our program. And this is a very robust market, as I mentioned to you. Only in the US, there is in 2021, the market value was at about 930 million. This is actually a very informative graph because it shows you the different areas that bone grafts and substitute grafts are used in order to stimulate bone formation. And the largest portion is the dark gray, which is the spine fusion market. And this market is where we are going to be. And we believe that given the efficacy and the safety of Oxy-133, we can have very significant penetrance into the market, mostly by competing with Infuse of Medtronic. Again, huge market with a rapidly increasing growth at a CAGR of 5.6% in the United States. So now the second problem that we're trying to solve, and this is a more robust area and a very huge unmet medical need, and that is fatty liver and Nash. So just let me describe to you what Nash is for those of you that may not be familiar with the disease. So the healthy liver in 20 to 30% of individuals, because of a number of different reasons, including obesity and diabetes, starts to accumulate fat. And that's when you have a non-alcoholic fatty liver disease. And the reason that it's called non-alcoholic fatty liver disease, because this can also occur with overconsumption of alcohol. But in this case, this is a non-alcoholic fatty liver disease. Currently, about 80 million people in the United States have this disorder. But it's not always, it's a lot of times it is benign, but in 10 to 30%, it further develops into what is called Nash. And what Nash is basically accumulation of lipids that also includes inflammation. And inflammation is bad because it starts to degrade the integrity of the liver tissue, which then eventually leads to cirrhosis, liver cirrhosis, where you have a lot of wounding, if you will, and a lot of breakdown of the liver microarchitecture. And eventually it can, in 30% of the cases, lead to hepatic carcinoma, which is liver cancer, which unfortunately, after a certain stage, Nash, when it goes to cirrhosis and carcinoma, there is no going back. And very often it ends up in the need for liver transplant. There are some ways that Nash can be reversed. There is some controversy around some of them, but clearly weight loss and exercise can help reverse Nash. But currently, and for the past decade, there has been a very robust activity in pharmaceutical sector to find a drug that can actually inhibit the transition of fatty liver into Nash and hopefully reverse it. And nothing has really worked, unfortunately. And this is a disease that is expected to keep increasing in the population by 63% between 2015 and 2030, just in the United States.
Why is that?
Well, that's because the population is aging, and unfortunately, obesity is increasing. Diabetes is still not under control, and it's just an unfortunate series of events that are resulting in this massive increase in the number of people who have fatty liver disease.
It's pretty disturbing. Yeah.
Yes, it's very, very disturbing. But unfortunately, what's disturbing is that there is no way to reverse it right now. There's no FDA-approved drugs, and there has been a graveyard of compounds that have been tested by many pharmaceutical companies with no hope, and everything has failed. What we hope to bring to the table is a completely novel strategy for going after NASH. And this strategy involves our lead compound, Oxy-210, again, another oxysterol that is orally bioavailable. It can be given as a pill or a syrup to patients. And what we published together with our UCLA colleagues is that it can inhibit hepatic fibrosis and NASH in mice that develop NASH in a fashion that is very similar to human NASH. And the reason that this is a novel strategy never tried before is that we actually are able to inhibit three main drivers of NASH, three signaling pathways in cells called hedgehog signaling, TGF-beta signaling, and toll-like receptor or TLR signaling, using a single compound, Oxy-210. That results in anti-fibrotic and anti-inflammatory effects. This has never been tried like this before, and I think this is why there is excitement, and this is why we received our first SBIR grant from the National Institute of Diabetes and Kidney Diseases, NIDDK, and we are hoping that the success of that phase 1 SBIR grant is going to lead into a positive review tomorrow of our phase 2 grant that will bring another 1.7 million in order for us to continue development of Oxy-210 for NASH. So this is really very significant in an area that has had no success in finding new molecules or any molecules for therapy. Just to give you a little bit of data, this is the mouse model that our UCLA colleagues used. This is a mouse that actually has elements of the genetic picture of the humans, and it develops NASH in a very similar manner to what human disease is after it is put on a high-fat diet, here called Western diet. And after 16 weeks, it has a lot of inflammation and a lot of fibrosis in its liver, and eventually these mice succumb to the disease itself. So what we were able to show is that by giving our oxysterol during a 16-week course orally, we could significantly reduce NASH and inhibit NASH. And here is just a few pieces of data. This is histology that you can see the red staining for collagen, which is the main fibrotic matter that accumulates in the liver during NASH. There is a lot of red when there is no oxysterol given, and there is much less red when the oxysterol is given. And this is evidence that there is a very significant inhibition of fibrosis, which is the basically the end result of NASH and demise of liver and its function. A pathologist scored these animals and found that there was a very significant decrease in the amount of fibrosis that is seen in animals that were given this oxysterol. And the fact that the oxysterol was given for 16 weeks with no safety issues adds to the fact that Oxy-210 is not only efficacious, but is also a very safe drug to pursue a clinical development with. So we also looked at a number of factors called cytokines that cause inflammation, and almost in every case that we looked, there was a very significant reduction in the amount of these inflammatory cytokines in the livers of these animals that received the oxysterol. And if you remember, I mentioned to you that Oxy-210 or the oxysterol has anti-inflammatory effects, and this is becoming a very interesting area for us to dive into, because this Oxy-210 is not only anti-fibrotic, but it also has very direct effects on inflammation. And that's why we believe that we have actually discovered very potent anti-inflammatory compounds that can help not only in NASH, but also in other diseases of chronic inflammation, for example, atherosclerosis and skin psoriasis and kidney disease and a lot of different kinds of chronic inflammatory diseases. So we're very excited about this. But again, all of this amelioration of the disease in these mice also results in preservation of the function of the liver. Here, this is a standard test that's done not just in animals, but also in humans, the alanine aminotransferase activity of the liver. And you can see that it is highly increased in control animals that received the high-fat diet, but also was reduced significantly. So there is a lot. So the lower the levels, the better the function of the liver. And as you can see, there is a very significant, robust lowering of the levels of this enzyme in animals that received the oxysterol. So this is very exciting. But the other thing that we found that I alluded to, and if I go back up to the slide that had the title, you'll see that not only hepatic fibrosis was affected by OxY210, but also hypercholesterolemia in these mice was affected. And the reason that this is important is because something that is now noticed more and more is that there is a parallel development of NASH, fatty liver, with atherosclerosis, which is when your arteries become blocked and circulation is impaired. And a lot of this is because of hypercholesterolemia and the fact that the oxysterol was also able to reduce cholesterol levels in circulation is something that we are pursuing. And we actually recently got news that our phase 1 SBIR grant by the NIH is going to be funded in order for us to study the effects of OxY210 in yet another chronic inflammatory disease, i.e. atherosclerosis. So this was a huge deal for us, and it's going to allow us to expand our therapeutic development program in the area of atherosclerosis as well as NASH. So let's go back here to where our funding situation has been so far. As I alluded to earlier, we were originally supported by about $50,000 in family and friends, which was in the form of convertible notes, followed by a seed funding, equity financing by a seed investor in the amount of $200,000. And this was about 6 or 7 years ago. And since then, we have been able to bring in 4 phase 1 SBIR grants from the NIH, 4 SBIR phase 2 and 2B grants. And this is something that Arthur alluded to earlier, and getting these grants is not trivial. It's very hard, and especially a lot of times your phase 1 grant fails, so it never gets to a phase 2. The only way that you get your phase 2 is if you have succeeded in doing what you propose to do in your phase 1 grant. So we've been very successful and very proud of this accomplishment, and we also received a $1.4 million grant from the California Institute of Regenerative Medicine about 2 years ago in order to go after developing a bone-targeted, orally bioavailable anabolic agent for the treatment of osteoporosis. So that work is still ongoing, but all of this has resulted into more than $8 million in non-dilutive grant funding. But now is the time that we need to go after more funding in terms of institutional funds. Again, this is mainly because it just takes so long for a successful grant to come in going the NIH route. And that's why we now are trying to raise $12 million. And we have decided to focus, as I mentioned before, on 2 of our most advanced therapeutic development programs, which is in spine fusion. We need another $3 million in addition to the $1.7 million that is currently supporting the IND enabling studies, because we need to perform a number of additional IND enabling in vivo studies in larger animals, as well as showing efficacy of OXY133 in spine fusion using primates, mainly monkeys, in order to find the dose in order to proceed into phase 1 and phase 2 human trials. So this is going to take a $3 million investment. And the fibrosis program requires $4 million in order to get us to the point of IND filing with the FDA for treating Nash. And this is to support the tox studies and PK studies. And hopefully, if our grant tomorrow gets reviewed and comes back with a favorable score, it will reduce the amount that is needed for the fibrosis program, because it will bring $1.7 million in non-dilutive grant funding. And we also would like to have enough assets in order to continue with the company efforts and our intellectual property matters for another 3 years, and that is for another $5 million. But so this is a $12 million raise, Series A. It does not take away from the fact that there are multiple other opportunities that require more funding. And currently, as I've been saying again and again, we rely on NIH grants in order to support those studies. For our viral infection program, the Japanese government has been very, very supportive, and they have basically supported the entire package of studies that went into showing the anti-COVID-19 effects of some oxysterols in our portfolio. We did publish this with our Japanese scientists in a peer-reviewed paper recently. And we recently have started collaborating with the NIH in order to demonstrate, again, the anti-COVID-19 effects of some of these oxysterols, again, all supported by the NIH directly. We have also started to and continue to look in the area of cancer, mainly pancreatic cancer and lung cancer. And we have actually selected a number of candidates that are anti-tumorigenic, and they need to be selected. And we need to come up with a lead compound in order to pursue a number of indications in the oncology space, namely pancreatic and lung cancer. We would like to put in more money into our osteoporosis program in order to pursue a much needed bone anabolic agent for bone for osteoporosis. This is, as many of you know, osteoporosis is still a very significant problem, despite having all of these anti-resorptive compounds, including Prolia and Timlos and all of the other ones that are either anti-resorptive or osteoinductive. And this is a huge medical need, and we believe that we can have something that is going to change the way that osteoporosis is treated. And of course, our inflammation program was just received the news that we will have an SBIR phase 1 grant that will bring about $300,000 into supporting further in vivo efficacy studies in the area of atherosclerosis, a very highly aggressive and serious vascular disease that is in need of new therapeutics. So again, the Oxysterol Therapeutics platform has resulted in a number of lead compounds, some of which we have actually done a lead selection, including Oxy-133 and Oxy-210. But in the area of oncology, we have Oxy-186, as well as other oxysterols that are inhibitors of hedgehog signaling, and they act very uniquely to other FDA-approved drugs that target this hedgehog signaling in a much more efficacious way. Oxy-210, which I mentioned, is being developed for fibrosis as well as for oncology. And we have a number of oxysterols in our portfolio that target hepatitis B virus and COVID-19 infections, and also Oxy-210 and Oxy-234, another potent anti-inflammatory agent, all of which has really resulted in our studies of our platform, Oxysterol Therapeutics. So this is our value proposition to those that may be interested in investing. We are working with proprietary small molecule oxysterols as part of this platform technology that can result in a number of different candidates that we haven't even gone over yet, through very specific mechanisms, mainly modulation of 3 signaling pathways in cells: hedgehog, TGF-beta, and toll-like receptor signaling. And these are highly involved in tumorigenesis, fibrosis, inflammation, and also for tissue regeneration. And as I have been saying again and again, there are many market opportunities that are available for MAX BioPharma as we investigate the Oxysterol Therapeutics platform more and more. So we have a number of patents that have been issued. Most importantly, probably for the audience, is the Oxy 133 patent that was published in 2017, or awarded in 2017, and also for Oxy 210, our other very promising lead compound that was published very recently and awarded very recently to MAX BioPharma, with a number of other patents that have been filed, either provisional or PCT, in the areas of viral infection and method of use for targeting fibrosis. And as I was answering Peter's question earlier, the only IP that we have that is exclusively licensed from UCLA is for Oxy 133. And that was originally filed in April of 2013 and awarded in 2017. But there is a lot of trade secrets that we hold at MAX BioPharma, which has and continues to add to the barriers of entry, because these molecules are not trivial to work with, and we are really the pioneers in studying oxysterols as drug candidates. So just to tell you a little bit about the other people that are involved, I have 2 distinguished scientific co-founders at MAX BioPharma: Michael Jung, who's a professor of chemistry at UCLA, and he's the inventor of Xtandi, a prostate cancer drug that some of you may have heard about. Dr. Bill Matsui, who was at Johns Hopkins and now is at the University of Texas in Austin, is a key opinion leader in hedgehog signaling associated with hematologic malignancies. So he's going to be very effective and important as we develop our oncology program involving the inhibition of hedgehog signaling in tumors. Frank Stoppenbeck is our director of chemistry. He comes from over 15 years of industry experience, and he's a co-inventor of Oxysterol Therapeutics, together with myself and Dr. Feng Wang, who is director of molecular and cellular biology. He's also a co-inventor of Oxysterol Therapeutics and has been critical in our ability to test the mechanism of action of our oxysterols that we identify for these different diseases. And we also have Gonzalo Payan, who is our director of finance and operations. He comes from over a decade of experience at UCLA in grants management. And in addition, we also have a very capable laboratory manager, Anthony Chow, who is in charge of the everyday management and maintenance of our labs. So we have a small team, but a very effective team. And of course, we have a number of scientific and business advisors, as well as consultants that we constantly are working with, as well as many experts in the CRO space that are helping us with many of our studies and development programs. So on that note, let me just end this by thanking you again for being part of this webinar, and I would be happy to answer any other questions that might have come up, and continuing this discussion maybe after this, if you're interested.
Yeah, well, of course, we'll make sure that everybody's in touch. A couple questions. Is it as obvious as it seems that the NIH and those type of non-dilutive grants are designed to fund therapeutics in this case because the feds are such big payers through Medicare and Medicaid, aside from just advancing innovation?
Mm-hmm.
So my question is it really their objective, because they pay out so much in Medicare and Medicaid claims that they're willing to, fund things that will decrease the amount of claims going forward?
Well, certainly, yes. And actually, one of the important goals of NIH is to support innovation in the area of therapeutics. So every taxpayer dollar that goes into these grants is with the idea of improving health in the general population, so that eventually it's going to benefit the government as well.
Yeah. So not with the intent of calling you out, but our dearest friend Iva has asked a question about your team as it relates to female content from female representatives in terms of advisors or others.
So we actually have a number of advisors that we work with who are female, and unfortunately, we don't have anybody on our team that is female right now. We did actually have someone that left the company. She was a very brilliant graduate student and postdoc at UCLA who was helping us with a number of our studies. But we have a number of people that are on our advisory board who are female, and also our CROs and consultants who are females. So yes, so we are very well balanced. And hopefully as funds come in, we will be able to bring in more female scientists to help us as our programs evolve, and as we add to the number of diseases that we're studying. We're going to need hands-on scientists, and hopefully it will be a nice balance of both male and female scientists.
And by the way, I know Iva very well, and she wasn't calling you out. She was just being curious. So,
sure. Yeah. Okay. That's good.
Yeah. And I don't mean to speak for you, Iva, but I think I'm probably okay. Talk a little bit, if you can, about, even though we know that this is your first, you're just starting this series A, how's the raise going so far?
Well, the raise has just started, and it's been only a few months. We have been reaching out to multiple institutional funds, both VCs and family offices, and discussions just are starting and continue. What is absolutely crucial is for us to be able to generate more and more data and de-risk our programs enough so that the traction is going to be more and more. And especially as we bring on board pharmaceutical companies to become our partners, I think it's going to allow for an even more effective raise that is going to be in the future. But it's new. It's very new to us. So
it just seems obvious also to me that if the brain trust, and let's just call it that, at NIH and those sort of places that are activating grants, that if they've gone through and voted with their checkbook, then that's got to be really good for you in terms of being able to have bragging rights and somebody else that really knows what they're doing, looking at your, not only just the technology, but the efficacy, right?
Yes, no, absolutely. So the peer review process is very important, because when you propose something, it is not just being proposed on its business merits, but also the scientific merits that it has. So these are all very savvy scientists who are involved, and key opinion leaders in your field who assess your application. And when you get, what we get is usually a summary statement that has comments and suggestions by reviewers. And we're always very happy to see the enthusiasm behind all of the comments that we receive from the reviewers of our grant applications. And again, this is why we have been so successful in bringing in multiple SBIR phase 1 grants that have led to SBIR phase 2 grants. So this has been, we're very proud of it, and we're very
You're
fortunate.
4 out of 4, right?
No, actually, we have applied for many other grants as well, which are continuously being revised and put back in. But yeah, we have had a good success rate.
No, I mean, you're 4 out of 4 in terms of going to phase 2. In other
Correct.
words, yeah.
Yes.
Yeah.
All of our phase 1s have gone into phase 2.
Yeah. Yeah.
Yes. So that's testament to the fact that we know what we're doing, and our plans are always good for continuing the development to the next stage.
Yeah. When we first started talking about this, you had mentioned to me that you hadn't really pitched this way for funding. And I will say you've done very, very well in an arcane subject. So, and I don't need to be nice to you. That's the truth. So,
well, thanks for being nice to me, Arthur.
Yeah.
But,
yeah, that's really helpful. So there are some other questions, but we're going to run out of time here. So we want to be kind to the audience here. Do you expect that you want $1 million checks, $2 million checks, $5 million checks?
The amount of investment can vary, and we can always talk about multiple ways to structure the investment. We're okay with equity financing. We're also okay with convertible notes if the amount of investment is smaller than the $12 million raise, because every portion of investment that comes in is going to allow us to make more progress and get closer and closer to the next inflection point for the company, which is filing with the FDA to initiate clinical trials. So yes.
Yeah, great. Well, we do need to wrap it up. Thank you very, very much for doing this. Jason said, great job. Super presentation. Right?
All right. Well, he's a big cheerleader for us. So
and he's a smart guy. So we know he's on your side. So well, thank you so much once again, and thank you everybody in the community for participating, and also your colleagues that were on listening in. And as I always say, thank you for spending with us the only thing you can't make more of, and that's your time. Till next time. Thanks, Farhad.
Thank you very much. Take care.
Bye-bye.
Bye-bye.